الله الرحمن الرحيم. طبعا أنا في البداية بحب أشكر حضراتكم على الدعوة الكريمة. يشرفني ويسعدني النهاردة أن أنا أكون بتكلم مع حضراتكم على الانتي نوفل كورونا فيرال فاكسينز، واتكلم ان الموضوع مش موضوع فاكسين ديسكفري بس الموضوع مهم جدا ان الفاكسينز دي تكون سيف وافكتيف، والسيفتي والافكتفنس بتاع الفاكسينز والchecking them actually needs a lot of efforts and a lot of work. It's a very long-term, tedious process. طبعا انا بشكر نقابه الاطباء جدا على برنامج التعليم المستمر اللي عاملاه، ومش كتير ابدا ان احنا نقضي ليله مع الاطباء الاعزاء اللي هم الفيرست ديفense لاين. actually if we're talking about immune system، يعني هقول ان الفيرست innate immune system defense line دلوقتي هو الاطباء المصريين الحقيقه، او الهيلث كير وركرز، لان هم they are the first to get exposed to the virus. وانا الحقيقه كل ما هيبقى في occasion اقدر contribute I'll be very happy. بشكر الاستاذ دكتور اشرف العدوي على المحاضره الشيقه جدا والvery nice introduction ان herd immunity can be one of the solutions, but to achieve it, as he said, you need to have at least 60 to 70% of the population exposed to infection. And this is first unrealistic. It takes long time. وفي نفس الوقت you would not be able to judge whether this would cause a lot of casualties ولا لا، زي ما حصل في السويد زي ما تفضل الدكتور أشرف وقال. أنا عايز أقول في عجالة يعني إيه فاكسين عشان الناس كلها تبقى اللي بتشوف معانا الويبينار النهارده، وانا I'm assuming ان في ناس مش سبيشالست بتشوف معانا الويبينار. الفاكسين يا اما الهول باثوجين يا اما component من الباثوجين تبقى structural ولا non structural، ودي able to induce immune responses، والimmune responses دي could protect the host against infection. يبقى مش اي immune changes في الهوست by default would mean that we have a vaccine. عشان بس في ناس are jumping to conclusions a lot and they think if they inject anything in a host and they induce immunity, it does mean by implication that they have a vaccine. أنا بقول لا يا جماعة، vaccine development it's really a process where you start with 100 candidates and you end up with a very few candidates. طيب التايب اوف vaccines ايه هم؟ اولهم and the most primitive one is a killed organism itself, the killed pathogen itself. If you can kill the pathogen or you attenuate the pathogen either by exposing the pathogen to irradiation or chemical treatment, you inactivate it. Or if you are purifying a protein or a molecule of the pathogen that is able to mount immune changes or to stimulate immune mechanisms or immune reactions. And this can be a native vaccine, or if you express one of the viral proteins in a recombinant form by just carrying a piece of gene, minal virus genetic material on a cloning vector or a plasmid. In this gene can either carry one of the antigenic determinants or several antigenic determinants, and accordingly you're talking about a monovalent or multivalent vaccine, a recombinant subunit vaccine. And recently or more advanced, the next generation vaccines are naked nucleic acids, are either DNA pieces or RNA pieces that are encoding under the mammalian expression machinery the proteins of the virus that might induce immunity. طبعا في حاجة very annoying, we have to stop it actually. واحنا بنتكلم لان انا كتير بسمعها في الميديا، بقراها في الجرايد وبسمعها في التلفزيون، ان الناس بتقول قريبا سيتم اكتشاف المصل الواقي. كلمة المصل الواقي دي كلمة خاطئة تماما. عايزين نفرق ما بين فاكسين وانتاي سيرا أو لقاح ومصل مضاد. اللقاح هو الباثوجين أو جزء من الباثوجين زي ما قلت، لكن المصل هو السيروم اللي أو البلازما اللي شايلة antibodies produced against infection and might neutralize infection. And one of a good example for that is a convalescent plasma from naturally recovered individuals. So when we talk about it's محدش يقول على اللقاح إنه هو المصل. كلمة المصل بالنسبة للقاح دي كلمة خاطئة تماما. الفاكسين جزء من الباثوجين، المصل هو السيرم وما يحتويه من immune molecules ضد الباثوجين. ده هياخدني للمرحلة اللي بعد كده في ترمينولوجي لازم نعرفها، الاميونوثيرابي. والاميونوثيرابي إذا استخدمنا أي immune molecules في العلاج. والimmune molecules can be monoclonal antibodies, polyclonal antibodies, cytokines or complement regulators. وده مختلف برضو عن vaccination or passive immunization using convalescent plasma. طيب, what is a good vaccine candidate? A good vaccine candidate حاجة بت-induce long-lasting immunity that can protect the host against infection, وفي نفس الوقت بتستيميوليت حاجة اسمها ذاكرة مناعية أو memory immune mechanisms، وما بتبقاش accompanied by undesired inflammatory responses. والundesired inflammatory responses دي حاجة زي cytokine storm اللي بتحصل في severe inflammation cases. وبعد immunobiologicals بعد ما حصل لهم discovery upon long term usage اكتشفوا ان هم ليهم side effects نتيجة ان هم بيستيميوليت بيستيميوليت cytokine storms upon usage، وعشان كده وقفوهم. فانا عايز اقول واحنا بنتكلم على الافيكاسي لازم تكون قدامنا على طول السيفتي بتاعه الاميون مولكيولز اللي بنستخدمهم فور ثيرابي. لازم برضو واحنا بنتكلم على الفاكسينيشن نتكلم على البيرسوناليزد فاكسينولوجي، ان ما نقدرش ناخد البشر كلهم one size fits all approach. ما نقدرش نimmunize الناس كلها زي بعض، لان actually speaking الهيلث ستيتس بتاع الناس كلها مش زي بعض. فالاميون compromised patients مختلفين عن الالرجيك patients، مختلفين عن الاوتو اميون candidates او subjects، واكوردنجلي ما اقدرش immunize الناس دول كاني immunize healthy individuals. ودي برضه should be taken in consideration while we talking about vaccine development and vaccination strategies. Getting to our story, النوفل كورونا فيروس. طبعا زي ما حضراتكم شايفين على الليفت هاند سايد ده شكل الفيروس، وهو ديكوريتد على الانفلوب بتاعه بالسبايك بروتينز. والسبايك بروتينز apparently are the most immunogenic antigens of the virus. Next to the virus, in the middle, you have the three-dimensional structure of the spike protein, and here you have the cleavage sites that activates the entry of the virus to the cell upon binding of the spike protein أو بروتين الشظية بتاع الفيروس للangiotensin convertase enzyme 2 اللي موجود كريسبتور على الخلايا الهوست سيلز بتاعت الهوست. Accordingly, we need to sort of intervene or interfere مع virus entry as first line blockage or virus propagation. So a good vaccine candidate would block or would interfere with the entry or would interfere with the virus propagation. طيب عايزين نقول ايه النوفل وايه اليونيك وايه اللي مخلي الكوفيد 19 او السارس كورونا فيروس 2 او النوفل كورونا فيروس مشكله. ان لما حصل الاوت بريك في فوهان في هوبي بروفنس في الصين وعمليه سيكونسنج للفيروس جينوم، لقوا ان السيميلاريتي بتاعه الفيروس ده للباتس كورونا فيروسز اعلى كتير جدا من الهيومن كورونا فيروسز زي الميدل ايست ريسبيراتوري سيندروم كورونا فيروس والسارس. وبالتالي ده خلانا نقف نقول احنا قدام فيروس ما هواش human virus, is more likely an bats related virus. لكن to complicate the story more, actually looking at the sequences of the spike protein, which is a rate limiting protein in the infection, لقوا ان في homologous recombination events في بعض البليسز في السبايك بروتين بتاع الكوفيد 19 مع البانجولين related coronaviruses. And this tells you that it's a mixed up virus actually, and very novel, in that might interpret why the jumping capacity or the spreading capacity of the virus is huge. Although the mortality caused by the MERS-CoV or the SARS is higher, but the spreading capacity of the COVID-19 is far much higher, and as a result, the overall mortalities caused by the COVID-19 is getting higher now. So altogether tells us that we were not prepared to face this virus. So what are the obstacles or the hurdles actually to develop vaccines against such viruses? We're left without biosafety level 3 to 4 facilities, either laboratories or animal facilities. من غير ما يبقى عندك high biosafety level laboratories or animal facilities, you cannot handle the virus, you cannot propagate the virus, you cannot try to modulate or manipulate the virus or interfere with the virus with antiviral agents to test them. You cannot infect animals with the virus after being immunized to see if the challenge infection could be blocked. In other result, if you're missing having the biosafety level 3 to 4 animal facilities or laboratories, you cannot really develop vaccines against the virus. To bypass this, actually, what we do is we clone viral genes, and we deal with viral genes as a representative antigenic determinants of the virus. Nevertheless, you're missing also a part, which is after having a candidate vaccine, you need to check the efficacy. You need to do challenge infection. Where would you do this? Unless you have a very high degree of safety, you are not allowed actually to do this. And this is one of the hurdles in developing vaccines in developing countries where those facilities are not well established. So what are the stages of developing vaccine for humans? So regardless whether we're talking about COVID-19 or other viral diseases or other pandemics, in the pre-COVID-19 era, actually, it's a very long story. It's a very time-consuming procedure to develop a vaccine for human use. So you need a laboratory stage where you discover candidate molecules that can serve as vaccine candidates. And for this, usually, people took from 2 to 5 years. And then you need preclinical evaluation for those candidates in animal models or in cell culture. And for this, you needed actually 2 years. And for the clinical studies, before making sure and before deciding that you have real vaccine, you needed to go through different phases, phase 1, 2, 3, regulatory phase and manufacturing phase. And actually, these are years in numbers of patients in very careful regulation. Otherwise, you're causing damage to the society, actually. So it is not only thinking of a developing candidate or protective vaccine, rather than thinking of not harming the society. And even if you get to the manufacturing in the production stage, it's economics. It's a lot of money you put in that. In this is actually, you need to establish unit GMP facilities, highly qualified facilities of very regulated conditions to produce vaccines at the same standards with the same reference procedure. And so you don't need to produce 5,000 vials of a vaccine in a way, in 10,000 in another way. You need to have one reference way, one standardized way for production of vaccines. In this, for this, you need to have well, reference units for production. In this costs money. So to make this long story short, actually, I made this cartoon, and this tells you that the discovery phase takes 2 to 5 years. In preclinical phase takes 2 years. Clinical development takes altogether 5 years to 7 years. And then you have the regulation and production takes 1 to 2 years. And the cost of this is $500 million for one vaccine. And I tell you, actually, so I have to get back to the previous slide. See, you start with 100 potential vaccines on the top, and you end up with one potential vaccine. So, so people has to think that you cannot tell people that I started with one vaccine candidate and I ended up with the same vaccine candidate. This means that you're not aware of how vaccine development is pain in the neck. So you need actually a lot of candidates, a lot of immunogenic molecules, a lot of antigenic determinants, and then you make combinations and you exclude some of them because they stimulate inflammatory responses, and you end up with one effective vaccine that is safe. But you cannot start telling people that you're developing 4 vaccines and ending up with 4 vaccines. So now, what makes this procedure fast? How can we make this procedure fast, actually? We need to work parallel on various vaccine candidates. And then we need to combine stages, and we need to find out seed money and share facilities to do this. And this money, I'm talking about billions of dollars. Actually, I'm not talking about millions of Egyptian pounds. I'm talking billions of dollars. And you need to develop global alliances, actually. You cannot do this alone. Oh, so some of this investment will fail. I know that you work with a lot of money on many candidates and you end up that none of them is effective. But at least you excluded that, and it's better than not doing it. And because the casualties and the extension of the pandemics and having new waves in the next years without having the appropriate herd immunity developed, as Professor El-Adawi mentioned, actually. So we cannot leave the situation like this. We have to work hard on vaccine development because this is the hope that people are relying on in future. What is the roadmap? We need to streamline the vaccine developing process. We need to raise funding and to fund as many vaccine candidates as possible. We need to create clinical trials insights across the world, so we do not rely on effective vaccine in one place, one geographical area in the world, because of the personalized and geographical factors. So you need to prove that the vaccine is effective. And by the way, I have to get back to this. Susceptibility of people, genetic susceptibility of people to infection is not equal. So people who receive infection and clear infection are different from those, even if they are not chronically, if they're not suffering from chronic diseases. You have healthy people who are susceptible, severely susceptible for infection and develop severe symptoms, and you have others who can cope with the infection and clear it. So you have to prove the efficacy of a vaccine in different places in the world before getting it into the market. And you need to build global manufacturing capacities. So you cannot build capacity alone in Egypt. You need to develop global alliances, actually, to develop enough that can be marketed, for example, in Africa. So I'm ending the presentation, getting to the end of my presentation with some hopes for you. So those are the companies across the world who are working now on phase 3 clinical trials of their candidates. On top, the Novavax in Maryland, and it is now going through a phase 3 trial that is applied or administered on 10,000 human in the UK. And you have Johnson & Johnson. And Johnson & Johnson is also having a clinical trial, phase 3, on 60,000 US humans or volunteers. The vaccine from Novavax is a spike protein of the virus that is carried on nanoparticles and mixed with M matrix adjuvant, while the Johnson & Johnson is a virus segment that is cloned into adenoviral vector. So you have a recombinant virus, recombinant adenovirus carrying a viral antigenic determinant, a coronavirus antigenic determinant. The Moderna company is also producing a candidate vaccine that is now tested in phase 3 clinical trials on 30,000 volunteers, and it is naked viral RNA. Pfizer, which is a company, a big company, actually with daughter and regional company in New York and Germany, is also producing RNA-based viral vaccine that is now in clinical trial in multiple countries. So they are not only doing the study in Germany or in the US, they are spreading the trial on 44,000 volunteers in different developing countries. The famous Oxford vaccine, in collaboration with AstraZeneca, which is also adenoviral vector-based recombinant vaccine, is now in phase 3 clinical trial on 50,000 volunteers in Brazil, UK, and United States. Sinovac and Sinopharm both are based on killed virus, and they are Chinese vaccines. Most probably the Sinopharm one is the one that we will use in future in clinical trials in Egypt. And they are in expanded clinical trials in Brazil, Indonesia, Bangladesh, United Arab Emirates, Peru, and Bahrain. So this was an overview on what is going on in terms of vaccine discovery against novel coronavirus, and hopefully soon we'll get one or two of those vaccine candidates that can be marketed all over the world to contribute to minimizing the transmission of the virus, together with the existing herd immunity that is not equal everywhere in the world. Thank you for your attention. And again, thank you for giving me the honor to participate today.